Did any of you ever hear of or see any of the television programs done by Steve Irwin, the crocodile hunter? Maybe a little bit before your time, I don't know. Anyway, Steve Irwin was killed by a stingray attack, but his show, when it was on, maybe 10, 15 years ago, was very entertaining. Well, that doesn't have anything to do with anything except that I saw his son on TV recently, and I couldn't believe how much his son looks like his father, acts like his father, talks like his father, and is almost just the spitting image of his father. It was very entertaining and interesting to see how the young man looked like and acted like and talked like his dad. Well, these things have been noticed throughout history, but nobody really knew what exactly was causing traits to be passed from parents to children and maintained in the family line. So that's what we're talking about when we talk about genes and heredity. It wasn't until about 1900 that a scientist named Johann Miescher uh, observed that it was some material in the nucleus of cells that contained phosphorus that seemed to be the molecule of genetics. He called it nuclein. We now know that stuff to be DNA. So when we talk about genetics, what we're talking about is DNA-ology. So maybe you recall that in the chemistry chapter, I showed you the 5 carbon sugar ribose, and I showed you how from ribose we can use a phosphate group and a nitrogenous base and come up with things like DNA. So let's have a look at that one more time just so that you can see what the DNA nucleotides are actually made of. Well, I hope that this looks a little bit familiar to you. So there in the center portion of the molecule, we have the 5 carbon compound. They are numbered 1, 2, 3, 4, 5. You would notice there off of that little ribose at carbon number 2, there is no hydroxyl group. If there was an OH at carbon number 2, then we would have RNA. Since there is no hydroxyl group at carbon number 2, it's called DNA, deoxyribonucleic acid, or if it's just the sugar, deoxyribose. From carbon number 1, we have 4 possible nitrogenous bases. They are adenine, thymine, guanine, and cytosine. If we had RNA, the 4 possible nitrogenous bases would be adenine, uracil, guanine, and cytosine. So if we're talking about DNA here, it's got to be A, T, G, or C, branching off of carbon number 1. From carbon number 5, we have the phosphate group, PO4. It is through the phosphate group linkage that we bind one nucleotide to the next. So that is called a phosphodiester bond. So one nucleotide binds to the next one by way of that phosphodiester bond through the phosphate group. Uh, Human DNA has 3.1 billion nucleotide pairs. Well, since it is double-stranded and we have 3.1 billion pairs, that would mean we would have 6.2 billion DNA nucleotides. It's a double-stranded molecule. And so we will look at how those two strands bind together in what is called complementary base pairing in the next slide. So this is a single DNA nucleotide. One would differ from the other only in the makeup of the nitrogenous base. So the nitrogenous base may be adenine, or it may be thymine, or it may be guanine, or it may be cytosine. So one differs from the other only in which nitrogenous base is present. So we could very easily abbreviate the sequence of nucleotides just by writing A, T, C, or G, because that's the only difference between the individual nucleotides. Okay, let's go to the next slide and just take a closer look at complementary base pairing between the individual nucleotides. Well, you can see that we have two different strands. The little pink boxes, that is the deoxyribose, the 5-carbon sugar. You can see the phosphodiester bonds extending from one nucleotide to the next. So we have two different strands of these. They are held together by hydrogen bonds. According to the law of complementary base pairing, anytime you have adenine on one strand, it must pair with thymine on the other strand. Also, anywhere you have guanine on one strand, it must pair with cytosine on the other strand. The adenines and thymines are held together by two hydrogen bonds. The guanines and cytosines are held together by three hydrogen bonds. So this is drawn out much like a staircase. The actual geometric design is like a spiraling staircase, so it kind of twists as you go up the stairway. So that is the findings of Watson and Crick. What is DNA made of and how is it laid out? So 1953, Watson and Crick offered to the world their model of, well, this is what DNA is made of, and this is how it's laid out, and this is how the two strands exist relative to each other. It is in the sequence of nucleotides that we have our individuality. Chimpanzees are about 98.5% identical to humans in their nucleotide sequences. Humans are 99.99% identical to each other, but just that 0.001% difference allows all of us to be perfectly unique in the order in which our DNA nucleotides are laid out. So that's what allows us to use DNA as an identification tool, say for a crime scene or to determine who's your daddy type of things. People have uh, nucleotide sequences which are unique to them. So unless you have an identical twin, then you would have a sequence of nucleotides which is unique to you and would not be found in any other person. All right, I have on the next slide a little close-up view of DNA as it kind of occupies the nucleus of a cell. Okay, inside the nucleus of our cells, we have 46 strands of DNA. Those strands are very thin and long. We call it chromatin when it's in that condition. The chromatin will condense down to 46 visible chromosomes during the stages of mitosis, but in the stage of, or the phase of interphase, it is in the form of chromatin. So that is a lot of DNA. Someone has said that if you put one chromatin strand against the end of the next one, it would amount to about 6 feet long. And all of that is crammed inside the nucleus of our cells. Well, if you're going to take that much DNA and put it in the small area of a cell, then it's going to have to wrap around and fold and zigzag and be all compressed together in order to fit inside of a small nucleus. So when we look at it up close, what we see, they're the orange colored things that look like a rope there. That is, those are the two strands of DNA. They wrap around proteins that are called histones. The histones together make up what is called a core particle. We divide the strand of DNA into units called nucleosomes. The nucleosome includes the DNA that wraps around the core particle plus the linker DNA that extends from that core particle to the next. So that is how the DNA is laid out in the nucleus. It's actually in a zigzag type of fashion so that it folds and compresses together so that we can fit all 6 feet of it inside the small nucleus of a cell. So these are the things that we find. There are, I think, 8 different histones that make up the core particles, and the DNA wraps around those histones 1.5 times before it advances to the next core particle. Okay, let's look at the next slide here and see what exactly takes place in the process of reading the DNA. This might be a little bit confusing at first glance, so let me explain a little bit of what you're looking at here. Remember that DNA is double-stranded, and the 2 strands complement each other. Anywhere you have a thymine, on the other side you would have adenine. Anywhere you have a cytosine, on the other side you would have guanine. So what is shown here, the strand to be transcribed is the strand in dark blue. We are not making a transcript from the strand in light blue. The strand in dark blue is the one to be transcribed. The message will be taken from that strand. We have a magic molecule. The molecule is called RNA polymerase. RNA polymerase has the ability to read the template strand of DNA. So the template strand that the RNA polymerase is going to read is the one shown in dark blue. So that sequence is TACTTCAAAATC. So that is the strand that we're going to read and transcribe. The molecule that does the reading and transcription is RNA polymerase. Anytime the RNA polymerase reads a T, thymine, on the template strand of DNA, that's the first nucleotide we have there, is thymine. Anytime it reads thymine, it will put into place the complement of thymine. The RNA complement of thymine is adenine. So notice on the pink transcript that we're forming here, the first nucleotide put in place is adenine. The next nucleotide to be read on the DNA template is adenine. Anytime RNA polymerase comes across adenine on the DNA template, it will put in uracil on the transcript. So our second nucleotide put in place on the RNA transcript is uracil. Go back to the DNA strand template. The third nucleotide to be read there is cytosine. Anytime RNA polymerase comes across cytosine, it will put in the complement to cytosine on the transcript. So the complement to cytosine is guanine. So our first 3 nucleotides in the messenger RNA transcript is AUG. AUG is also known as a start codon, which tells the ribosomes this is where you start. That's why it's called a start codon. It is also the codon that calls for the amino acid methionine. Remember, ribosomes synthesize proteins. Proteins are made of amino acids. So the DNA, by its command TAC, that command is sent as a message to the ribosome as AUG. The AUG is read by the ribosome or translated by the ribosome as this is where I start, and the first amino acid I need is methionine. Go get me some methionine. So the units of 3 nucleotides in the messenger RNA are called a codon. So the ribosomes read 3 messenger RNA nucleotides at a time. In other words, they read the transcript codon by codon, 3 units at a time. When all is said and done, the ribosomes will read the message sent by the DNA by way of messenger RNA and will create a polypeptide, a sequence of amino acids, which become our proteins, in response to what the DNA called for, which was transferred to the ribosome by way of messenger RNA. So, if your DNA nucleotide sequence is unique to you, which I have already asserted that it is, if your DNA nucleotide sequence is unique to you, then the proteins that you synthesize will be unique to you. This is why you can't put foreign proteins in your body. Your immune system will reject them. So the body is kind of a holy temple. We only want self here. If you put foreign proteins into your body, your immune system will react against them. So we make our own proteins in response to our own DNA nucleotide sequence, and those are the characteristics that are our traits. Now remember, we got half of our DNA from our daddy, we got half of our DNA from our mama. Well, they got theirs from their parents, and so the traits continue in the family line because the nucleotide sequences are passed on from parents to children. So this is how we understand genetics these days. It is much more complicated than this, but for a course in structure and function of the human body, offered in a brief period of 5 weeks in a summer term, that's just going to have to do. If you want to know the details, you're going to have to take a class in genetics. I think I'm pretty safe in saying that we all started out as one cell. Well, that one cell underwent mitosis. So after mitosis is completed, we have a new cell. So let's start with that. So start there on the right where it says growth and normal metabolic roles. That phase of the cell cycle is called G1. What goes on in the G1 phase of the cell cycle is growth and normal metabolic roles. The cell is simply doing what the cell was born to do. If it's a muscle cell, well, it contracts and relaxes. If it's a nerve cell, well, it conducts action potentials. So the cell is simply doing in the G1 phase what the cell is uniquely qualified to do. So it's called the first growth phase, or G1. Many of our cells do not advance past G1. They simply go into their normal metabolic roles, and they will not ever advance to a mitotic division. We have stem cells to replace them when they die. So they don't undergo any mitosis. The stem cells that produce your skin cells, or the stem cells that produce your blood cells, the stem cells that produce your nerve cells when you were in the uterus, that's where those cells came from. So they go into a G1 growth and normal metabolic roles, and that's where they stay. They don't undergo mitosis. But for those cells that do undergo mitosis, then we're going to advance through the cell cycle here. So let's go from G1, that phase of the cell cycle where the cell is just doing what the cell is supposed to do, secreting hormones or absorbing nutrients or whatever the cell does as its normal metabolic role. Well, we then go from that for those cells that do undergo mitosis. We go into the phase where the DNA has to be replicated. So we have 46 chromosomes. Those 46 chromosomes will double themselves. So we have at that point what we call 92 chromatids. It is 46 chromosomes times 2. So we end up with 92 chromatids because the DNA has doubled itself. So we call that phase of the cell cycle the synthesis phase because we are creating 46 additional chromatids of DNA. The DNA is being duplicated. Okay, advancing from the synthesis or S phase of the cell cycle, then we go into the phase called G2. In the G2 phase, for those cells which do undergo mitosis, what happens in G2? Well, the cell continues in its normal metabolic roles, doing whatever it was created to do, but we have to make enzymes and we have to duplicate organelles. We have to increase the volume of cytoplasm because we're preparing for mitosis. In order for one cell to become two, well, then you're going to have the things that can divide in two and still give you two normal cells. So things have to be duplicated. You've got to increase the number of organelles. You've got to increase the volume of cytoplasm. You've got to make the enzymes that are needed for mitosis to take place. So that takes place in the G2 phase of the cell cycle, which is preparatory for the upcoming mitosis, the division of one cell into two cells. Now, one of the reasons that I teach about the cell cycle is because there are some interesting things that can happen here. In the mitotic phase, where we divide one cell into two, and the DNA has to be equally divided between the two daughter cells. Because that is a hazardous activity, the DNA can become damaged anywhere in the cell cycle. We're exposed to mutagens all the time. Ultraviolet light from the sun damages DNA. Certain viral infections can damage DNA. Exposure to certain drugs and chemicals can damage DNA. Just the process of mitosis itself, the DNA can come out damaged. So we have, it is not shown here in this particular illustration, but between G1 and S, we have a checkpoint where the checkpoint is analyzed by a protein called P53. We want to analyze the DNA to make sure that it's still normal, because we're going to take that DNA and duplicate it. And from it, we're going to make two cells rather than one. Well, we don't want to do that if the DNA has been damaged. So the P53 inspects the DNA and has the power to fix it. Now, what if the DNA is so badly damaged that it can't be repaired? Well, the P53 should destroy that cell, because that cell is headed for mitosis. And what you're going to have is double damage if it completes mitosis. Well, we have also, before the mitotic phase between G2 and mitosis, we have another checkpoint to inspect the DNA to make sure that it is as it should be before we create a new cell from it. Otherwise, you're going to end up with two abnormal cells. Two abnormal cells become four, four become eight. Pretty soon you have a collection of abnormal cells. That is called cancer. So these checkpoints that we have in the cell cycle are designed to inspect the DNA. If it is abnormal, then we want to stop it at that point. If we can't fix it, then we need to stop it. So this is how cancer begins. We have damage to DNA. The damaged DNA advances through the cell cycle, ignoring the checkpoints, undergoes mitosis, and where you had one abnormal cell, now you've got two. Once you have two, you're going to have four. That's called a tumor. So the cancer cell ignores the checkpoints or bypasses the checkpoints, and pretty soon the cancer cells are so numerous and they're reproducing so quickly that the immune system cannot keep up with them, and you end up with metastatic cancer. So the cell cycle is an interesting thing. So familiarize yourself with it, and make sure you tell little sister about it. I just can't imagine how big her eyes might get when you tell her about your knowledge of the cell cycle that you picked up as a student at UCA. I suspect she will probably get up and try to enroll at UCA immediately. Next slide, please. In prophase of mitosis, those long spaghetti-like strands of chromatin condense down to chromosomes. The chromosomes can be seen. Well, we have 46 chromosomes. Half of them came from mama, half of them came from daddy. Well, we can pair them up. So when we pair them up, we end up with 23 pairs. So look at pair number one. Pair number one of those came from daddy, the other one came from mama. They are identical to each other, they just came from two different people. So they are chromosome pair one. Chromosome pair two, one of those is paternal, came from daddy. One of them is maternal, came from mama. Well, they're identical. They are the same chromosome, they just came from two different people. So this is how your chromosomes are laid out. The laying out and illustration of the chromosomes is called a karyotype. So when we have all 23 pairs together, the last two are the sex chromosomes. The sex chromosomes in this particular individual, both of them are X. Therefore, the individual is female. The presence of a Y chromosome always is a male. This person doesn't have a Y chromosome, has two X chromosomes. So this is how we study the chromosomes and check them for any abnormalities that might be present. Now, the genes are a segment of the chromosomes. We have about 20,000 or 25,000 genes, a section of DNA that commands for the synthesis of a particular protein or a related set of proteins, is called a gene. The genes are in the chromosomes. So each chromosome has genes. Some chromosomes have more genes than others. So we would say that some chromosomes are gene rich, other chromosomes are gene poor. Just all we're saying there is that some chromosomes have more genes than others. All said and done, the smart guys have determined that about 98% of our DNA does not get involved in protein synthesis. So we say that that DNA is asleep. It is dormant. We only use about 2% of our DNA in the process of transcription and translation, therefore protein synthesis. So we have a lot of DNA inside of our nucleus that we really don't know what it does. It appears to be asleep. It's not transcribed by RNA polymerase. No transcript is made from it, therefore no proteins are synthesized. So we don't really know exactly what 98% of our DNA is doing. I suspect that it is doing something. We may not know what it's doing, but I suspect that the 98%, which is determine, well, it must be asleep. It doesn't appear to be. I suspect that it probably is doing something. We do know from some experimentation in the 1970s that certain genes which appear to be asleep can be awakened. So when we have that sort of thing, genes which should be asleep are awakened by some environmental factor. Oftentimes, the result is cancer. So we have certain viruses, which when they infect the nucleus of a cell, they can cause sleeping or dormant DNA to awaken. And when that sleeping or dormant DNA is awakened, then proteins are synthesized in response to it. In other words, it gets transcribed. Well, we may not want those proteins to be transcribed, so we can end up with some strange disorders, one of which is cancer. So those sleeping genes, which when they are awakened, cause cancerous processes to be initiated, those are called oncogenes, cancer-causing genes, which may be awakened by chemicals, may be awakened by radiation, may be awakened by certain viral infections. All right, so we know what the normal karyotype for a person should look like. So we would look at this under a microscope and say, well, this is what these chromosomes ought to look like. Well, what if you see something different from this? Well, then you've got a chromosomal abnormality, and most of those are incompatible with life. Some of them are survivable, but most chromosomal abnormalities are incompatible with life. So let's have a look at a few abnormalities in the human karyotype. So here we have three different karyotypes. Look at the first one on the left there. We have 23 pairs. That's fine. That's a total of 46 chromosomes. The abnormality present here at pair number 21, there is an additional chromosome. So that is called a trisomy. There are 3 chromosomes at position 21. That is not good. We don't need any departure from the normal arrangement. The normal arrangement is 46 chromosomes paired up in units of, or in total of 23 pairs. Here we have a third chromosome where there should only be two. Well, that is Down syndrome. It is a survivable aneuploidy because chromosome 21 is gene poor. The more gene rich a chromosome is, the less likely the person is to survive an abnormality there. So because chromosome number 21 is gene poor, we have an abnormality. We have an additional chromosome present there that should not be there. It is a survivable condition. So Down syndrome results in moderate mental retardation along with some other anatomical and physiological characteristics. The life expectancy of a person with Down syndrome is only about 50 years. There are some cardiac abnormalities, there are some intestinal abnormalities that compromise the life expectancy of a person with Down syndrome. So when a child is born, a chromosomal analysis can be made. If you see 3 chromosomes at position 21, then you have a Down syndrome child. Okay, look at the middle karyotype shown there. Whoa, something is wrong here down there at pair number 23. Those are the sex chromosomes. It's supposed to be a pair. So at position number 23, where we would have either XX or XY, what we got there is just one X, so there's not a pair. So what we're missing here is the second sex chromosome. So since there is not a second sex chromosome, the only sex chromosome that's there is the single X chromosome, then what you have is a total of 45 chromosomes. Well, 45 is not the number we're supposed to have. We're supposed to have 46 chromosomes. So this is Turner syndrome. Since there is an X, but there is not a Y, then you have a female. The presence of the Y chromosome commands for maleness. Well, there's no Y chromosome here. There's no chromosome at all there. So you have a total of 45 chromosomes. So that would be Turner syndrome. This also is survivable. The reason it's survivable because what's missing is the Y chromosome, and the Y chromosome is the most gene poor of all the chromosomes. There are very few genes on the Y chromosome. So if it doesn't show up in the offspring, the condition is survivable. It is far from ideal, however. Turner syndrome patients generally have moderate mental retardation and a variety of anatomical problems. They are sterile. They cannot reproduce with just the one X chromosome that's present. So there are a lot of terrible things that go with Turner syndrome, depending on how severe the anatomical problems are and how severe the mental problems are. Some Turner's patients can live independently. They can do certain jobs. So it is similar in some respects to Down syndrome as far as the abilities of the person goes. But the Turner syndrome patient will always be infertile. All right, the third karyotype shown here, well, let's analyze it for normality. Uh-oh, look there at position 23. We're supposed to have chromosomes in pairs. At position 23, we've got two X's and a Y. Well, that means we have a total of 47 chromosomes. The human number for chromosomes is 46. So these are aneuploidies, variations from the normal number. The normal number is 46. If you have anything other than 46, then you have an aneuploidy. In this case, the aneuploidy occurs at pair number 23. So what we have are two X chromosomes and a Y. The presence of the Y chromosome commands for maleness. So this is a male individual, but the male individual got a double dose of the X chromosome. So this is Klinefelter syndrome. You have a male who is sterile. There are some anatomical abnormalities. They generally are very tall and gangly. They may experience development of breast tissue that is called gynecomastia, and they do have some compromise in their mental abilities, but it is classified as mild retardation if it's present at all. So the Klinefelter person is male, but the male is sterile and unable to reproduce. So these are three aneuploidies resulting from chromosomal non-disjunction. So in the process of meiosis, the preparation of the reproductive cells, the sperm and the egg, if chromosomes don't separate properly, then you end up with a non-disjunction and therefore an aneuploidy. Two others that you might be wise to, in position 13, if you have a trisomy 13, so instead of position 21, which is Down syndrome, if the trisomy occurs at position 13, that is called Patau, P-A-T-A-U syndrome. That is horrific. Uh, Chromosome 13 is not exactly gene rich, but it's not exactly gene poor either. So if we have a trisomy at position 13, the anatomical and the mental development is profoundly arrested. Most Patau babies do not survive beyond 1 year. The problems are too devastating. A different aneuploidy, another trisomy, can occur at position 18. When we have a trisomy 18 child, we have, again, severe mental problems. That is called Edward syndrome. There are some Edward syndrome children that do survive up to maybe age 20 or age 25. Most Edward babies, life expectancy perhaps 1 year or less. So anytime the chromosomal abnormality is in a location where there are many genes, the chances of survival goes down. So it's only aneuploidies or non-disjunctions that occur at gene poor areas that are survivable. All right, next slide, let's talk about some inheritance patterns. Well, I hope that you recall from your general biology class, there was a man named Gregor Mendel, an Austrian monk, who in the 1860s experimented with his little garden flowers and garden pea plants to understand patterns of inheritance or the transmission of traits from parents to offspring. So he used his little peas and his flowers, and he decided that, there are two types of genes. So we call the two types of genes alleles. That's just the name we give it, two types of genes. Well, they are alleles. Well, one is a dominant allele, which means if it's present, it will be expressed. That's why it's called dominant. So if the dominant allele is present, it will be expressed. The other type is a recessive allele. So genes come in two types, dominant allele or recessive allele. If we have homozygous dominant parents, so both parents in this illustration here would be capital D, then 100% of the children would come out capital D, capital D. Capital D is the dominant trait, so dominant allele, so that trait would be expressed. Well, it becomes interesting when you have heterozygosity. So the parents that are shown here, the father has the dominant allele and he also has the recessive allele. The mother offers the dominant and also the recessive. So both of these parents are heterozygous. They have the dominant allele and the recessive allele. When they have children, the mathematical probabilities that go with their children are shown in the Punnett square. So the capital D from daddy, the capital D from mama, well, you got no choice. The child has both dominant alleles. The two heterozygous children, well, you've got a small d from daddy, you got a big D from mama, well, you've got a heterozygous child. So the chance there is 50-50 that the child will be a carrier of the trait because of the recessive allele being present. It will not express, it expresses the trait because the dominant allele is present. Okay, the 25% or 1 out of 4 possibility that a child is born with both recessive alleles, small d. Not always, but usually the recessive allele is the troublemaker. Now, that is not always true. There are some exceptions to that. So, the recessive allele is expressed only if it is in the homozygous state, small d. If the dominant allele is present, it will be expressed. Usually, the dominant allele is the healthy allele, but not always. So in this case, we have a 25% or a 1 in 4 possibility that the offspring will have homozygous recessive, little d. And the recessive trait usually, not always, is the troublemaker. So we have then, when we mix heterozygous parents, the possibility of a pathogenic combination, such as small d here. This is what we would see in something like sickle cell anemia. This is what we would see in cystic fibrosis, where you have children born with both recessive alleles. So this is why genetic screening of parents, if there is a family history of certain diseases, then 2 prospective parents should be screened and therefore counseled as to what the probabilities are that their children may actually be born with the homozygous recessive condition, which usually is the troublemaking combination. Not always, but usually. So this is how Mendel, or this is what Mendel did using his garden peas and his flowers, and identifying these patterns of inheritance which can be used to anticipate the condition of offspring, and that's where it becomes valuable. Now, the Mendelian pattern is still very accurate and very useful, but there are some more contemporary modifications that have been made since the time of Mendel. And so we see that, well, it's not quite as clear cut as Mendel might have wanted it to be or might have said it to be. So we do have some variations in the Mendelian pattern that you need to be wise to. So let's go to the next slide and I'll tell you about those. Well, according to the Mendelian pattern, if we have a red flower, 2 dominant alleles, big R, and we match that with a white flower, which is two recessive alleles, then 100% of the offspring would be heterozygous. They would be big R, little r. According to the Mendelian pattern, if the dominant allele is present, then the dominant allele will be expressed. Well, all of the children have the dominant allele. They have the big R. So we would expect all of the children to express the dominant allele, which is red. The red is dominant, the white is recessive. So since all of the children have the dominant allele, then we would expect all of the children to be red. Not so. So this is called incomplete dominance, and Mendel observed this himself. How is it that the dominant allele is present, but the flowers are not red like they should be? They come out to be a combination of the two traits, so the flowers are pink. So that is called incomplete dominance, and we see that in a lot of human traits also. Another variation here is codominance. When we have a parent who has the A blood type, and we have another, well, let's say a mother and a father. So a mother is type A, the father is type B. Well, we have children that will show both. So that's called codominant. So the blood type of the children has both A and B proteins on the red blood cells. So we call that codominance because both traits are expressed in the children. One doesn't dominate over the other. They both dominate, so the child is both blood type A and B. The condition of pleiotropy. This is where one gene can affect the expression of more than one trait. Well, the two things that I have cited here are known to be pleiotropic problems, alkaptonuria. What we have in the disease alkaptonuria, one gene can cause the eyes, the whites of the eyes, to turn black. At the same time, the same gene can affect the functioning of the kidneys, and the urine is black. And there are also some other physiologic problems that go with alkaptonuria. But what we have found is that one gene affects the expression of more than one trait. So something like the coloration of the sclera of the eyes and the effect that it has on the kidney, well, that's two different things, but it's caused by one gene. Also in sickle cell anemia, the thing that we associate with sickle cell anemia is the effect that it has on the shape of the red blood cells. But there are also other abnormalities present in the sickle cell patient besides just abnormally shaped blood cells. Well, it's one gene, but there is more than one manifestation of the trait in the person. So that is called pleiotropy, not something that Mendel would have known anything about. In the case of polygenic inheritance, what we have is one trait that is affected by many different genes. I suspect, and don't hold me to this, I'm not a geneticist, but I suspect that when we understand genetics better, we will find out that basically every trait is probably polygenic. There's more than one gene that affects the expression of our traits. So that's what polygenic inheritance is. Things like eye color, skin color, certain cancers, a tendency toward alcoholism, an inclination toward mental illness, all of those we have identified more than one gene that plays into whether or not a person will express those types of traits. So those are slight modifications from what Gregor Mendel offered to us. All the same, the Mendelian pattern is largely accurate and trustworthy for counseling purposes. Okay, at position number 23, that's where we have our sex chromosomes. Recall that I told you the Y chromosome is gene poor. It is the most poor of all the chromosomes as far as, well, how many genes does it have? Alternatively, the X chromosome is gene rich. So we have then a male child. The male child picked up his X chromosome from his mama. He picked up his Y chromosome from his daddy. So we have a mama who has a recessive allele that she passed on to her son. The Y chromosome doesn't have an allele, it's gene poor. So even though the trait is recessive, it is expressed in the male child because the Y chromosome doesn't have an allele to outshout it. If there is another allele that's dominant, which would be present in the female child because she would have two X chromosomes. So even if she inherited the recessive allele from her mother, then she would have gotten the dominant allele on her X chromosome from her father, and the dominant allele on that X chromosome would outshout the recessive allele on the chromosome she got from her mother. Well, the male child doesn't have that luxury. He picked up the recessive allele from his mama. He picked up the Y chromosome from his daddy. The Y chromosome doesn't have the allele. So in that case, a recessive allele is expressed because there isn't anything there to outshout it. So that's what happens in X chromosome linked recessive traits. The mother passes on a recessive allele to her son. Since he doesn't have another X chromosome to outshout the recessive trait, then it ends up being expressed in the male child. It doesn't happen in the female child, it happens in the male child. So examples of that, red-green color blindness is an X-linked recessive trait. Duchenne muscular dystrophy, an X-linked recessive trait expressed in males. And there are about 5 different types of hemophilias. Something like 3 of them are X chromosome related. So they are expressed in male children. The male picks it up from his mother's X chromosome. Since the Y chromosome can't outshout it, then the male child exhibits the trait of hemophilia. So there are several others that are X chromosome linked, but those are just a few just to hang on to. All right, I think that's all I've got for this show. Sorry to be so brief and have to abbreviate it so much. If you would like to learn more, the biology department at UCA does offer courses in genetics.